So for this next section, I want a discussion on basic science. Obviously, the line between basic and translational is very blurry, but because we will be talking about translational next, let's try to keep this focused more on things like how GABA receptors function just in the healthy condition and what needs to be understood there. And then for the translational, we'll talk more about variants and drug development models like mouse models and sort of that sort of thing. So
I just want to ask, because I'm trying to educate myself a lot here, and this has translational implications, but basic science. It's basically how important are the actual levels of the different subunits? So if you have a little bit, does it kind of restore the pentamer well? If you have too much, do you start to get over incorporation? I'm asking this like my expertise in gene therapy, and so the dosage sensitivity is a big issue for us. And so I'm just wondering how big of a concern that is, or if the multimeric complex kind of self-regulates the over or under expression that it really is in one particular subunit.
So we have done some experiment for kind of computation for each subunit. So overall, we think in the brain means alpha 1, beta 2, beta 3, gamma 2, because gamma 2 is like one to anchor the receptor at the synapse. But the alpha 1 and the beta 2, they are essential for receptor assemble to pentamer. So the first, if you only have alpha 1, beta 2, they can form pentamer with either 2 alpha, 3 beta, or 3 alpha, 2 beta. These both exist. So with that's from document and work. So we found out you change the ratio, you see. And for gamma 2, I think Andrew, you still remember that. With 0.313, we actually get a 0.3 and 1 get a very similar current, get a very similar surface expression. But when you have a 3 microgram, you have a huge amount of ER stress and stuck inside the ER, even a wild type. So this why is very sensitive to the dosage.
Also, we did the catamer work, which is, catamers are a little dicey because you're constraining the proteins. But when you get to the higher doses of gamma-2, we found that you get phage receptors, where you have 2 gammas that we don't normally think happens in living brain, and it had a disrupted pharmacokinetics. So it's a good question.
It's very good, very useful.
And now Sijikowski can do a different result, can do different titration.
Yeah.
I think she used a different.
We have worked the large undertaking lab to figure out this. So I think you ask a very important question, because as Dr. Anna Grant said, fake receptor. So those are not physiological at all. So this is why you have to be very careful, do not overwhelm the system. So that's why you do not overwhelm the quality control. So now you have a very good conservative quality control system inside the cells built in. But if you give too much, you can overwhelm the system. So this is why you have to within the range. Stay within the range.
Maybe you could say a couple of words on what's possible today with gene therapy. I just heard that there was a CRISPR-Cas therapy that was approved.
That translational will be more of the next section. I just want to be sure that we get the time to talk more about like the basic understanding of GABA-A receptors instead of having it all taken over by translational, because I think that will be the tendency of the discussion.
Okay, well we can talk about that later. I mean, there's a wide variety of ways that a mutation or a variation can cause defective GABA-A receptor function. One of the ways would be a dominant negative effect, where you have a defective subunit that damages the activity of the receptor in the membrane. Is this something that you see happening, Addie, in your experiments? Do you have the variant subunits be trafficked into the receptors in the membrane, and that they therefore have reduced function because of the dominant negative effect?
We have done a lot. So when we talk the dominant active effect, so it's not just the assembled into the receptor, the functional channel itself. We also talk about the gain function in the cellular toxicity. So basically, they will entrap your wild-type allele inside the ER. So the and reduce the function. So when we talk about the function, we talk about overall function or like the function of a channel per se. So if you have a traffic defect, if you incorporate, you have an overall reduction function, but maybe in some condition, your receptor itself have a better affinity. So that's why you have to see overall level or the channel itself.
But genetically speaking, are they always autosomal recessive?
Some of them are dominant. Yeah, many of them are dominant. Many
Heterozygous. All the patients are heterozygous. Most are only a few are.
They're all dominant, and the variants that are like nonsense variants, they tend to be more likely to be familial, whereas variants in the missense will tend to be more likely to be de novo.
With beta 3 will cross all the GABA-A receptor.
Oh, across all.
So somewhat related to this topic that we haven't touched on for the basic science side is we can't treat all cells like they're the same population. So with a nice introduction on neurons or skeletal neurons, but effectively targeting inhibition, it would be ideal if you could target subunit compositions that were unique to inhibitory or to excitatory neurons over inhibitory neurons. And I think there's still a little bit of a gap in our knowledge to what the native receptors are in those different cell types, which would be really beneficial as we start to get to the translational conversation. But I think learning a lot from the NMDA field is that knowledge can be very powerful when you want to target specific cell types expressing specific types of GABA receptors.
Very good point. I think we actually for GABA-A receptor, we have very good knowledge in this area. Also, I have a very good expert, Dr. Maria Clemencia Hanadas from Roche. She has studied over 20 years. And we have a whole team at Vanderbilt who study GABA-A receptor. We have good idea.
Speaking also for our colleagues that have Steve Moss is unable to be
It's
here.
a godfather.
He's
Yeah.
done a lot of
Yeah.
work along these lines. And
Absolutely.
There's and criteria that represents that work. But I think one of the limitations technically has been getting enough starting material of inhibitory neurons to see what are the native receptor conformations in those cells.
Very important. Yeah.
They're really a piece of knowledge that we currently don't have.
Also, the association with the disease phenotype. For example, we know like beta 2 delta is more enriched in cerebellum, and alpha 3, beta 3 is more enriched in thalamus. All those knowledge is definitely essential for translation.
That's an excellent point. As one of my questions is about the different brain regions. What do we still need to understand about other brain regions and their role in the in just regular functioning and disease pathology?
Well, the thalamocortical system is also one that's particularly believed to be involved in absence seizures, which you heard is present in some patients with these instruments. So, John, you and Art and others are doing out of work on characterizing the GABA receptors in the system.
I mean, for the most part, most cells express a variety of subunits, but there are some relatively clean areas. So like in the sensory thalamus, it tends to be alpha 1 beta gammas and alpha 4 beta deltas. And the NRG, it tends to be alpha 3 beta gamma. And in parvalbumin, yes, they have other GABA receptors, but as far as I know, you might know there's none of the like it. I think that they're the only cell type that express alpha 1 beta 2 delta, which has unique properties. And so I mean there's reason for all. Yeah, there is some limited information out there about
Could the field use an effort for an atlas, a mouse atlas to start of receptor distribution, not by cell type, but by brain region? We know the cell type distribution by brain region already, but the receptor subtype and the pentamer composition for brain region, I don't think we
there has been quite a bit of work done. In fact, I took my slide out because I didn't think I'd have time to show it, but it's actually in the deck later on. I show the various alpha subunits that are expressed in different brain regions. But I'm not sure that's necessarily going to get us to where we need to be. I think we need to understand kind of the fundamental mechanism of the disease. And one of the things that really is surprising based on the work that Phil discussed and what we actually talked about yesterday in the SLC, the GAB transporter mutation epilepsies, where you actually have increased amounts of GABA and you also get seizures. So it becomes paradoxical. We've always in the field felt that you increase GABA, that should cure your epilepsy problem. Well, it turns out not to be the case either in the transporter defect or in the SSADH defect, or you mentioned in the GABA transaminase defect where you're seeing seizures as well, maybe even greater than for the SSADH. So it's a complicated story.
What I want to add is that I think it echoes what Phil just talked about. It's actually the brain is developing. So when you have a gene that's mutated and when you develop from a very early stage and where brain is actually different. So the trajectory of that brain development is very different. So how do we understand that mutation cause what kind of pathology? I think that kind of animal modeling or cell model is actually right.
Yeah, very good point. So I have a look at the beta-3 workout animal model. So at the beginning, you see clear reduction for beta-3 from postnatal day 1. But at 7 months old, you see huge upregulation of beta-2 subunit. And the beta-3 actually, in a mutant animal, is a similar level with the wild type. So I think at a certain point, some genes, you don't need anymore. Like it's your plateaued. But the others, a compensatory mechanism kick over. Maybe in clinical, the symptom we see, for example, in microneurologics, some phenotype is actually associated with the increased beta-2 instead of loss of beta-3. So that's why for gene therapy, if you restore beta-3, maybe in long run, you will normalize the increased beta-2. So that system is very dynamic.
And then the GABA receptors that express during periodontal life are entirely different than those expressed on both prolonged collagen and pseudoatlas.
Your problem with looking at the subunits, they can change rapidly. GABA receptors turn over in like 10, 15 minutes all the time. And so with combinations of the different subunits can be altered very rapidly. And that depends on your activity, so like epilepsy and allergy states can be altered the neuroactivity, which causes phosphorylation, which alters the target and stabilization of all different subunits. Also, neurosteroids. So we see that in cortex as hippocampus in our proteomic studies, that alpha-4 is just alpha-4 beta-3. But then you add on a neurosteroid like allopurinol, then you get the delta. So you're getting changes in subunit expression based on hormonal levels as well. And so it's very complicated to say this subunit is expressed in this cell. You've got to specify at what time.
What time?
At what
condition?
Are they asleep? Are they active? That could potentially change subunit combination as well. Very, very complicated.
I also want to add something to what Andre and Caddy were saying since they're on development, in that, and what we were talking about before about compensatory changes that happen with the mutation in the GABA-A receptor. So also GABA is involved in a lot of developmental events, and also GABA is an excitatory input, as Kieran was talking about. So the complexity comes into when you have mutation, especially in the subunit that's expressed early on. How is that affecting whatever developmental processes that GABA might be involved in? It ends up actually being really complicated because you've got the mutation in that, plus then you have changing chloride concentration, and you have the compensation that's happening in the brain, trying to compensate for the deficit in the subunit, and then you also have the changes in development that occur because of the mutation in the subunit. So it gets really complicated.
It does. I would just say that it's also an opportunity, right? For the fact that all these different, it gets super complicated. Right? One mutation versus another, one subunit versus another. But if we can understand the underlying mechanisms beneath them, maybe that gives us a way to be able to treat more than just one type of variant. Maybe maybe you could have one treatment for several. Maybe there's another treatment that will work for several others. But at least there's, or maybe understanding the underlying mechanism helps you to be able to focus more on, have a better target to focus your time and energy on a treatment target that might be relevant to bodily and the variants. So I would just point that out, that the understanding the underlying mechanisms of compensation may allow us to either prevent an issue or to utilize those mechanisms as treatments.
Absolutely, totally agree. Because back to Stephen Gray's gene therapy, we have developed a mouse model with overexpression of gamma-2 gene. I was trying to cross with beta-3 mutation and gestalt mouse for a long time. Have not done yet. So I think about if we replace like a mutant beta with gamma, for example, maybe some genes like you get a gene therapy available, maybe can be used for multiple subunits. That's possible.
Well, but that overexpression mouse, does it have any adverse phenotypes?
No, we have not seen any in wild type. So this one, last night I was in the Dravet Syndrome Foundation too. I asked them about the question. So they use CAD, the alien vector, in a wild type SNY. It's in wild type with 2 good copies already, and then with this gene therapy over a year, no phenotype. So somehow, if the cells can regulate the threshold of protein expression, that would be ideal. And that.
I mean, that goes back to my original question. Is I'm just wondering, does the cell have enough compensatory mechanisms that if you overexpress only one subunit, how bad would that be?
That have to be which subunit. For example, if you do beta-3 as a homomer, it can go to the cell surface, at least in HEC cells, in a cell model. But if you do alpha-beta with very little gamma, probably your cell will adjust. You will intentionally, probably somehow make a gamma maybe more efficiently incorporate into the receptor.
So if we're talking about gaps here, well, and I think you might have had a comment, but I'm wondering.
This reminds me a long
time when mouse band regenerate, God is blessing the fights ability, and then they be so gray over expression that we hardly saw any fan of that. So we never published that.
Well, so if we're talking about gaps, then is this something where our understanding of this is mostly theoretical, or do we have sort of hard examples of instances where subunits have been overexpressed, and it is or is not bad?
A better question maybe is there a beta-3 or a alpha-1 duplication syndrome?
Yeah.
Have a look. But there's plenty of genome sequencing you should be able to find if that's pathogenic or not.